Today we're going to discuss the large-scale in vitro production of monoclonal antibodies and how Sagittarius products can refine your upstream and downstream bioprocess. Now you probably have a targeted monoclonal antibody for IND nomination, but you're looking to optimize the process and produce 200 liters of your candidate for phase 1 or phase 2 studies and possibly for future commercialization. Now for today's presentation, we'll be looking at Sagittarius portfolio and how the products can be streamlined the bioprocess workflow and provide platform solutions to meet the 200 liter scale-up goal. Now for today's presentation, we'll be covering the following topics and agenda. Now the first step is step 1, which is the cell line development and optimization. In this step, it's necessary to isolate a stable, high-producing cell line in terms of quality and functionality. Now, to meet those requirements, high throughput screening of specificity of antibodies against known antigen is necessary. Now, this allows throughput assessment of a large number of clones for multiple factors in a single experiment. Now, to meet this demand, the AMBER-15 cell culture can provide 24 or 48 automated parallel bioreactor analysis on a 10 to 15 mil scale. Now, the system makes early high throughput screening optimization easier because each microbioreactor vessel has an integrated single pH and dissolved oxygen sensor, propeller steering, ports for liquid additions, and a gas sparging tube. Now this step determines which clone to take forward for cell expansion. Cell lines must be stable and maintain production over time and even over several generations. And this leads to step 2, which is the process development. Now the development process looks at the characteristics of a cell culture environment. Each cell line responds differently to a given in vitro production environment. So in the process development phase, a design experiment or DOE is a valuable methodology to organize, plan, execute, and statistically evaluate different variables for the environment. Sagittarius AMBER-250 high throughput perfusion system provides the tools to perform parallel operations to provide the optimal basis to efficiently realize a series of experiments to screen to achieve high cell density. Now the system combines 12 to 24 single-use bioreactors, which provides a fully automated bioreactor platform, similar to the AMBER-15 cell culture, but on a 100 to 250 ml reactor volume scale. Now this allows process developers to adjust multivariable factors such as temperature, pH, and culture time to understand the monoclonal antibody's growth rate and concentration over time. So this leads up to step 3, which is media selection. Now this step goes in tandem with step 2 in optimizing the process development. The culture medium has an important impact on both the yield and quantity of the monoclonal antibodies produced. Now for an in vitro system, the need to reduce the animal use and decrease the presence of contaminating foreign agent antigen is vital. Hence, process developers should focus on serum-free medium composition, buffer, feed, and oxygen and glucose uptake as well. Now, the initial pilot and manufacturing scale productivity can only be as good as what is achieved during the early development stages. And with the AMBER-250 workstation system, it can provide the capability to continuously monitor and control critical parameters in real time. It also can be extended to feed and sampling options to enable a more effective scale-up, and we can demonstrate the equivalent process performed in comparison to laboratory and pilot scales. So most of what we discussed so far is about refining the cell line for monoclonal antibodies, the environment, and the medium conditions to isolate a particular cell line seed for our monoclonal antibody scale-up. Once a particular seed cell line has been isolated, we can start the bioprocess scale-up towards the 200 liter target. Now, Sagittarius offers many solutions and paths to ensure a successful 200 liter scale-up with precision and quality. Now, as we continue to explore cell expansion and scale-up, media preparation on this scale becomes more important, and that leads us to step 4. So selecting appropriate mixing equipment can dramatically affect the quality and efficiency of the bioprocess environment. Now, Sagittarius FlexSafe Pro Mixer offers a flexible and intelligent single-use mixer and can maximize throughput and production. Now, the FlexSafe Pro Mixer can be equipped with mixing bags ranging from 50 to 3,000 liters, which makes achieving the final 200-liter scale quick and easy task. And each bag has inline controls that can monitor pH, conductivity, and temperature, which can all meet PAT and cGMP requirements. In medium scale to large scale production, it's important to have tight procedural environmental control to minimize loss to the system and microbial contamination. Now, the FlexSafe system provides a flexible single-use sterile environment from mixing, filtration, storage, and transferring. So as we start with our cell seed, we can rapidly expand the cell line with the Amber 250 high throughput perfusion system to automate the whole entire liquid handling system in the 250 ml reactors. With the perfusion setup, which enables media exchange in a bioreactor, we can achieve high cell density, which can reduce several expansion steps. Now, after completing expansion on a 250 ml scale, we can transfer the reactants to a 2-liter flex bag with a 1-liter max working volume. And once viable cell density has been achieved on the 2-liter scale, we can transfer the culture to a 20-liter flex bag to continue the cell expansion. Now, for the setup, the Sartorius Biostat B universal benchtop controller will make automating cell expansion at this volume straightforward. Now, this controller is capable of working with various other bioreactor configuration, but for today, we're going to be looking at the Wave Mix Rocker with FlexSafe RM bags. Now, the FlexSafe RM bags are ideal for volumes from 1 liter to 200 liters with a max working volume of 0.5 to 100 respectively. Ideally, the single-use flex bag makes cell expansion easier and more cost efficient. Now, this single-use technology offers numerous advantages over stainless steel and glass bioreactors, including enhanced facility flexibility, time-saving setup, cleaning, sterilization, and reduced overall capital investment. In general, the seed train expansion with FlexSafe bags are routinely used in bioprocess as they decrease contamination risk, which is most important. So when the 20-liter cell density has been achieved, we can venture out towards production development scale at the 50 and 200-liter scale. Now, the Biostat STR and Flex STR bags make single-use bioreactor operation reliable, safe, and easy at this scale. Now, with the Biostat STR bioreactor, we can incorporate advanced features such as an integrated spectroscopy port, ATF cell retention device, biopack control tools and software, and many other analytical tools. Now, this helps ensure quality, consistency, and precision. Now, after your 200-liter bioreactor has reached critical cell density and mass, it's time to recover, or in this case, harvest the monoclonal antibodies. And this is where we begin the downstream process. Now, the first primary recovery process is done through centrifugation coupled with depth filtration, where you remove the cell and cell debris from the cell culture broth. Additionally, you can clarify the cell culture supernatant that contains the monoclonal antibodies. After centrifugation, depth filtration are employed to further clarify the cell culture broth from insoluble impurities. Depth filters rely on both size exclusion and various absorptive binding methods to improve separation. The filtration process is generally scaled linearly from 10 microns to submicron size to about around 0.2 microns or smaller in most cases. Both Sartorius K-SEP and Sarto Clear depth filters can achieve primary recovery processes. The K-SEP offers continuous single-use centrifugation that improves recovery of both solid and liquid while selectively removing small particulate impurities. Now, the Sarto Clear depth filters offer a practical and easy cassette exchange to adapt to any filtration application to maximize product recovery. Now, before proceeding to the chromatography, the selection of an appropriate buffer plays an important component of the downstream process. Now, buffers are responsible for maintaining purification conditions, protein dimensional structures, and stabilizing protein while preserving its functionalities while protecting the protein from pH variations. Sartorius Flex-Act modular system is capable of expedient buffer prep time and quality while combined with Ceratopore 2. Now, for monoclonal antibody purification, this step usually involves protein A chromatography, followed by 2 or 3 subsequent chromatography polishing steps. Now, the chromatography column continues to be a favorable monoclonal antibody purification technique, and there are many options for chromatography medium, especially from Sartorius. Now, in step 10, we start looking at low pH viral inactivation. Now, the Flex-Act system can be reconfigured for virus removal through automated pH adjustment. Now, this automated low pH virus inactivation ensures precision virus clearance in the subsequent chromatography and polishing steps. Now, the first polishing step we'll be looking at is ion exchange chromatography. Most monoclonal antibodies will include at least one ion exchange chromatography step, and most antibodies will have a basic isoelectric point. And cation exchange chromatography can be used as an initial capture step and is frequently applied as a polishing step. Now, this step helps the removal of byproducts and processes related to impurities and viruses. Now, subsequent polishing steps may include a flow-through anion exchange and hydrophobic interaction polishing steps. Now, for a flow-through anion exchange chromatography, it's basically designed to remove residual impurities such as host cell protein, DNA, leach protein A, and a variety of viruses. Now, this polishing step, Ceratol Blend Q, is a good choice from the Sartorius Affinity Chromatography portfolio. Now, for a flow-through hydrophobic interaction polishing step is an efficient means of removing a large percentage of aggregates and impurities from the monoclonal antibody product. And for this application, Ceratol binding phenyl is recommended. Next is the virus filtration. After 3 polishing steps, you have the virus filtration. Now, this step removes a broad side of viruses depending on the pore size of the filter. Now, since mammalian cells are used in the manufacture of monoclonal antibodies, mammalian-derived products may contain less than 1 virus particulate per million doses. Now, the inclusion of virus removal and inactivation is an integral component of the product safety assurance strategy. Using a Flex-Act modular system with viral SART HF gamma sterilizer filter option will improve the final virus filtration throughputs. In step 15, the ultra and diafiltration cross-flow filtration. After the virus filtration, tangential flow filtration, or TFF, can be used to concentrate and diafiltration. In this case, the fluid that passes across the filter, also known cross-flow, tangential to the plane of the filter surface at a constant pressure. This process separates the permeate from the retentate. Now, the permeate includes everything that passes through the filter, buffer included, is sent to waste. Now, the retentate retains our targeted monoclonal antibodies and is recirculated back into the supply tank. And now, during this process, a new buffer solution is added to the feed. In effect, the buffer solution is washing with a new buffer solution, which helps remove unwanted salts, while controlling the final concentration of the product. Again, this process and step can be achieved using the Sartorial FlexSafe cross-flow modular setup. And now we get to the final stage, which is the bulk drug substance filtration. Now, after the ultra and diafiltration process is complete, the monoclonal antibody product is routed through a final micron filter. Now, for the final bulk drug substance filtration, Ceratopore Platinum offers a 0.2 micron filtration, which maximizes the yield and low protein absorption with a modified hydrophilic surface. Now, after this step, the monoclonal antibody product can be sent through a further downstream for final fill finish and freezing and packaging. And now that concludes the overall development process for monoclonal antibody upstream scale up to 200 liters and the downstream purification steps needed. So basically, no matter where you are in the development phase, Sartorius carries a wide assortment of tools, equipment, and knowledge to meet any demand. Now, if you have any questions about the upstream or downstream process, please feel free to contact me, and I hope you enjoyed this presentation.